Hello, my name is Sillitoe. I work at UCL, and I'm going to be talking about using CATH to predict the structure and function of novel protein sequences. So what is CATH? Well, CATH is a protein structure classification database. Essentially, we take PDB structures from the PDB and we chop them into structural domains. And then when we have these structural domains, we group them together into what we call superfamilies, where there is evidence that they are related through evolution. So we take structures from the PDB, we chop them into structural domains. Domains that have a common evolutionary ancestor are grouped into superfamilies, and these superfamilies are organized into a hierarchy, which basically puts these superfamilies in the structural space. And the hierarchy is where CATH gets its name, so class, architecture, topology, and homologous superfamily. And this classification is based on expert curation, which is guided by automated algorithms. So we can get an awful lot of information from the structural data, but we also know that we can get lots of information from the protein sequence data as well. So Gene3D is a sister resource of CATH that allows us to add sequence data into the information we get from structure. Essentially, it predicts the location of CATH domains on all known protein sequences. It does that by using sequence comparison techniques such as Hammer3, where we build a library based on the sequences from CATH structural domains, and then we scan protein sequences, all the known protein sequences, past this library to predict where these CATH structural domains lie on these sequences. So if a given protein sequence has extra annotations, such as a uniprot sequence might have gene ontology annotations or enzyme commission annotations, that might be very useful. And we can use this process to inherit that information to bring in sequence information, but also functional annotation information into the existing data in CATH. In terms of the update and release cycle of CATH, CATH is a continually updated resource. So we have a bunch of automated scripts that assist us to bring data in and to push that data through to annotate, to make curation decisions. If they are very, very similar to decisions that have already been made by a human, then we can automatically inherit those annotation decisions. So these are continually updated all the time. So we provide up-to-date annotations for everything in the form of daily snapshots of what the very latest amount of data is. But we also provide these extra releases, these CATH plus releases, where we freeze the data at a particular point in time, and then we add lots of extra data on top of that release. And this is where we put a lot of our own research into this data. So this is like generating three-dimensional models, generating alignments, functional annotations, and so on. So I'll talk about some of that extra data that we add into a CATH plus release. But it's useful to know that both of these exist. So if you're only after the very latest assignment of domains and superfamilies, then you can get that from our daily snapshot. So just to summarize what are the data and tools if you're interested in using CATH from that perspective. So CATH itself can provide the very latest annotations in PDB, and we provide that via daily snapshots, which essentially contain domain boundaries and the superfamily classification. And that they include automatic and manual annotation, and there may be minor changes between what you find in this daily snapshot and what you find in the CATH Plus release, because we add in lots of extra curation steps when we generate the CATH Plus to make sure that we're happy with all the decisions that have been made, but also we add lots of extra analysis and data, so alignments, functional annotations, and so on. I'm going to be mentioning more about that later. There are data sets there for non-redundant structural domains, if that might be useful for your own work or algorithm benchmarking. And we also provide lots of these tools on GitHub as open source tools for things like structure comparison, superposition, resolving sequence scans and domain architectures, and so on. The latest CATH Plus version, which is 4.3, has just over 150,000 PDB structures. It has just over 500,000 structural domains, and they've been classified into around 5,000 superfamilies. 4.3 was work mainly done by our CATH curator at the time, Dr. Natalie Dawson. Not all superfamilies in CATH are uniformly distributed. So it turns out that actually lots of superfamilies are very small, and a small number of superfamilies are very large. So a small number of superfamilies contain lots and lots and lots of diversity of structure and sequence and function. So this plot just gives a highlight of in the green sectors are showing the 100 most populated superfamilies according to the number of sequences that these superfamilies contain. So there are around 5,000 superfamilies in total, but the 100 most populated superfamilies actually contain more than half of all of the known sequences. So the largest superfamilies can encompass a huge amount of sequence structure and functional diversity. And this is really important when we want to use these superfamilies to start making predictions, particularly about function. So just to kind of give an idea of what that looks like, these are three example structures from a given superfamily in CATH, and that contains some diversity. So the ancient core, conserved core, can be seen highlighted in green, and this is the same right the way through these structures, and that's essentially what brings them together into the same superfamily. But you can also see that there are some structural embellishments around that core, and those structural embellishments also make changes to the function. As you can see, the functions for all these three domains are different. And the structural embellishments could change an active site, they could change residues around an active site, they could change the binding site. There are many different ways that these embellishments can change the function. And that's one of the advantages of this CATH as a resource, is that you can kind of see the evolution of how sequence changes structure, changes function. But there is certainly lots and lots of diversity within some of these superfamilies. So in order to take into account this functional diversity, we need something that's a bit more specific than grouping these domains just into superfamilies. And this is where these functional families come in. So these are functional families or fun fams within CATH. The idea is that all of these 6 domains that you see here in front of you, all of these 6 structures are in the same superfamily in CATH, and the common core again is highlighted in blue. The 3 on the left, they all do a particular function. Highlighted by the EC term 1.1.1.1. So these will all catalyze a particular substrate, and you can see that they will have a particular sequence pattern that highlights which positions are really highly conserved and control that particular enzyme function. And the 3 domains on the right-hand side, despite being the same superfamily, might have slightly different changes to the structure. They essentially do a very similar function, but on a different substrate, hence the EC classification 1.1.1.2. And those slight changes in the substrate will make changes to the sequence and the conservation patterns of those sequences. And that's essentially going from the superfamily level down to individual functional families or FONFAMs. So how do we do that? Well, we can start with all the domains in a particular superfamily, and then we can cluster them together, starting with the most similar sequences first, and then we keep on clustering them iteratively until we build a tree that contains all of the sequences. And the job then is to find a way of cutting this tree to give us the largest clusters possible without combining different functions, combining clusters that do different functions. And of course, we won't know what the functions are in all of these cases, so we have to design an algorithm that is able to try and do this automatically. And how we do that is by the idea of these specificity determining positions. So we kind of try to identify these SDPs by looking at, so when we're trying to decide whether 2 clusters should be kept apart or whether they should be grouped together, we look for positions that are conserved across all of them in the resulting alignment. And this might correlate to a position that's very important for the overall fold, or the overall structural fold. But there might also be some positions in the alignment where they're very highly conserved, but in one group they're very highly conserved as one particular amino acid, and in the second group they're very highly conserved as a different amino acid. And in these cases, it might be that that position is useful for determining the specificity of a substrate, or helping to decide which substrate the structure is useful for. So we use this kind of information to decide when to keep clusters apart and when to bring clusters together, and to try and calculate these functional families. Now, the amount of structural data we have is increasing, and the amount of sequence data we have is increasing even more. So part of the recent work in CATH was to bring all of these calculations into a pipeline that will work on the ever-expanding world of sequence data. And this is work started by Tony Lewis and then benchmarked by Shioni, and they are brought onto an HPC pipeline by Nico in the group. So in terms of actually using these functional families, how they're useful outside. So the easiest place to start is the CATH website. And just to also mention some of the other changes that have been made to the CATH website recently. So one of the jobs that we have been working on is using lots of manual curators to provide names and descriptions for lots of our superfamilies. So we now have a much more rich description of what makes a superfamily. So hopefully this should be useful to people using CATH. But also another feature that we've added is that in addition to these structural superpositions of all the structures in CATH, so this kind of allows you to see the type of variation for representative structures within a superfamily. We've also added these beautiful figures from the 2D PROTS resource, which shows the just a schematic diagram of the secondary structure elements of all of the structures within a superfamily. And it allows you to see the conservation and the differences that are present in a given superfamily. In terms of the funfams, we can the easiest way is just to cut and paste your protein sequence into the web page, and then when you search, you will get a list of matches to the individual structural domains. You'll also get a list of matches to the functional families, funfams. And then when you look at the actual matches, you will see that for your query protein, you will see the region that has matched a particular funfam in CATH, and whether that funfam has an associated representative structure in 3D, and whether it's got any annotations for enzyme activity. And by clicking on one of those, you'll be able to see that your query sequence, or certainly the region that has hit a particular funfam, that region will be aligned to the rest of the sequences in the functional family, and you'll be able to see the conservation patterns for that funfam and how that maps onto both your query and also the representative structure, if there is one, for that funfam. So hopefully by mapping the sequence patterns onto your query sequence and mapping that onto a structure starts to give a rich picture for your query protein. And there are also additional functional annotation information available for the funfam itself in terms of gene ontology terms, EC terms, and so on. If you wanted to tie this into your existing pipelines rather than manually checking web pages, then there is a documented API for the funfam searches, and also there's documentation on how you can take this offline to do more extensive scans over entire genomes, for example. So it's all well and good saying that we can make predictions about function from sequence, but how do we know that this works? And I'm going to be skipping over the details here, but just to let you know that this is a core part of the work in doing these predictions is trying to benchmark them and check that they work. So we think that these, if the funfams are working well, then they should be good at predicting gene ontology terms, i.e. functional adaptations from sequence. They should be good at predicting the location of important residues, and they should be structurally coherent, i.e. all the structures within these functional groupings should be very similar. And I can just essentially show that each of those 3 points is correct for these funfams. So in terms of predicting Go terms from sequence, the best way to prove whether they're working or not is using the CAFA experiment. So this is an international blind study that essentially throws out lots of protein sequences to the community. They ask the community to provide their protein prediction, the Go term predictions, and then they allow these sequences to gather annotations through experimental labs. After a certain amount of time, the organizers of CAFA will judge the predicted Go terms against the actual Go terms to see how closely these predictions are. These charts that I'm showing here come from CAFA 3, and the essentially low numbers show that the predictions are good, and the Orengo funfams have been consistently ranked top 5, top 3, top 1 for both molecular function and biological process in all of the CAFAs, including the most recent one. So we know that they work pretty well. In terms of predicting location of important residues, so we can look at the alignments, the sequence alignments of these functional families, and we can look at the conservation patterns of these alignments to show which residues in each sequence are very, very highly conserved. And if one of these sequences comes from a known structure, then we can map these conservation patterns onto the structure to see whether these match other features that we might know about that structure. So, for example, if this structure is an enzyme, then it might have an active site, and we would expect that active site to be highly conserved. If this was involved in binding to another domain partner, then we would expect those residues to be highly conserved. So this is another way that we can check that the FunFams provide cluster sequences together whose conservation patterns match what we already know in other experimental data. And lastly, we can look at the structural coherence. So when you look at the structures within a particular FunFam, we can see whether we can measure whether these structures are structurally close or not. So we can see that the FunFams, just by design of bringing together those the sequences based on these kind of functional constraints, actually also bring together structures that are also close. And one of the ways that we can measure that is by looking at the RMSD, which is a measure of structural similarity. So high RMSD means things are further apart in structure. Low RMSD means that structures are very similar. And if you look at all of the structures within the superfamily clustering, so these are the kind of the broad superfamily clustering, then we can see that the average RMSD is higher, i.e. the structures are more diverse. And if you just look at structures within the FunFams, you can see that the RMSD is much lower because the structures are much more conserved. And indeed, moving from the FunFams that we created in 2013 to the FunFams we created more recently, the RMSD goes lower and lower because the clusters are getting better and better. Okay, so another point was an application of us using these FunFams to make predictions about the H2 spike complex in SARS-CoV-2. This is a paper that was published in Scientific Reports, where essentially we modeled the spike protein H2 complex from 215 vertebrate species, and we calculated the changes in the energy of this complex caused by mutations in each of those species, all relative to human H2. So then we correlated these changes with the known data from known infection data, so which species were known to be able to carry COVID-19. And then, long story short, the results suggested that the SARS-CoV-2 can infect a broad range of mammals, but very few fish, birds, and reptiles. This has implications for these animals potentially acting as reservoirs for the virus. So that was the motivation for this work. So essentially, this started with a structural analysis of the residues involved in this key binding site. This is the key pathway of SARS-CoV-2, and either these residues were in direct contact with the binding, and those residues in direct contact are in red, or they were residues that were predicted to be important with this binding, and they were predicted to be important by the FunFam analysis, and they're shown in blue here. So essentially, we calculated the physicochemical disruption of all of the residues that were deemed to be important in and around the binding site in different species. And we could see that when including the surrounding residues predicted as important by FunFams, these kind of measures of disruption correlated very well with experimental data of whether the species could be infected with SARS-CoV-2. There's lots more detail that I'm not covering, but just as an overview of one of the applications that we have used for these FunFams. All this data has been put onto a web resource. So one of the applications here was COVID-19, as discussed. Another application was looking at cancer mutations in this CATH-Fun-Bar web resource. And this allows you to browse data in terms of the annotations made by FunFams, but also to look at the residue level effects on structures as well. So that just leaves me to say thank you to everyone in the group who has made all of this work possible, and our group leader, Prof. Christine Arengo, and all of the funding bodies. Thank you very much for listening.